everyone. We're here for the first science session. I'm Liana Orlando. I'm the VP of Research at Cure Duchenne.
And I'm not Liana.
You're applauding because he's not Liana? No, so we're thrilled to bring you this first session, which is preserving, protecting, and regenerating muscle. And this is a great session to start all of the science sessions out because it's the most applicable, I think, to everybody. Do you want to say some?
No, I think this is great. These are standalone therapies, and that's the way they're designed. That's the way the clinical trials run. But they're also designed to be add-on to other ones, such as gene therapy or exon skipping, where you're putting dystrophin back in again. So look at it in that context, and it'll give you some guidance about how this is important to you.
And the other thing is these therapies, because they're not trying to restore dystrophin, they work no matter what your mutation in the dystrophin gene is. So I think they're more broadly applicable than some of the other therapies that we'll hear today. And the last thing I want to say is we've got a great lineup. And this is a good problem to have. There's so many approved drugs and clinical trials which are going on that we forced all of the people you're going to hear from to speak for only 8 minutes. So this is the point for you to recognize who they are and figure out who you want to find in the exhibitor showcase later. They're all happy to answer questions and have you learn more.
Very good.
Excellent. All right, so do you want to do the first up?
Oh, is that for me?
Oh, it's me, actually.
Okay.
If you could put that slide of the speakers back up, we'll just give you a little overview. So first up, we're going to have PTC, which has the FDA-approved drug Emflaza, which is a corticosteroid that some of you may be familiar with already. We'll get some overview from them, followed then by Catalyst, which is talking about Agamree, which is a dissociated steroid, which has some of the sort of favorable properties of corticosteroids, but less side effects.
Next will be ITF. This is another approved drug. Duvizat is what's called a HDAC inhibitor, a histone deacetylase. And it's got quite complex biology. You may hear about some of it today, but that complex biology leads to multiple effects of the drug. It is the first approved non-steroidal anti-inflammatory agent that will also affect inflammation, fibrosis, and various other components. It's been marketed by ITF for a couple of years now, so you'll hear some of the latest data that's been coming out from non-clinical use.
And then switching gears from the FDA-approved drugs, then we're going to hear from Edgewise, which has a different approach. This is a small molecule which is trying to prevent the damage that occurs to muscle fibers with contractions. And the other thing that's interesting about Edgewise is they are in clinical trials not just for Duchenne muscular dystrophy, but also for Becker muscular dystrophy.
Yeah, and I'll just add, like the Becker one, I was talking to Joanne last night, it constantly is reassuring to me to see that Becker patients who've got no real treatment options now have got something that's meaningful to them, and the data that's coming out from that is really leaning heavily into a drug that's the first drug that's likely to be approved to treat Becker. Following that, there'll be 2 companies talking about cell therapy. The first will be Capricor, and Capricor has just finished their phase 3 trial. That there was the HOPE-3 study. That's a very significant long trial with over 100 patients, placebo-controlled, met primary endpoints, secondary endpoints, and it's up for approval this year. We're delighted to see that. They've got a PDUFA date, which is a date set by the FDA for review of August of this year. So the world is watching very carefully and very closely as the company's leaning into the latter part of the year for an approval on that drug. That's going to be followed by Secretome. Secretome are a much earlier phase, again, another stem cell focused company. They're in the clinic right now for heart failure. That's heart failure with preserved ejection fraction. But that there, as you recognize, heart failure is a significant problem in Duchenne, and the company are moving over to recruit this year their first clinical trial in Duchenne, focused on heart failure in those patients.
And then we're going to hear from Satellos, which is another completely different approach. They're trying to target muscle stem cells or satellite cells. And, you know, we recognize that we can do all we can to try to protect muscle from damage, but it would also be really nice to boost the systems to help muscle regenerate. And so that's what they're aiming to do with their therapy. And we'll hear a little bit about their phase 2 clinical trial, which is currently ongoing.
Excellent. And the final one is a company that many of you may not have heard about. That's Grünenthal. Grünenthal are a well-established German company, marketed products for over many years, and they've got a real focus on a business model where they've made a lot of products available in Europe. They're launching into the US right now with a brand new mechanism. This is a small molecule oral drug that they call a Seagram. It's a selective modulator of the glucocorticoid receptor. It is not a steroid, but the small molecule targets the steroid receptor. And it's designed to have the beneficial effects of a steroid, very potent anti-inflammatory, without all the negative metabolic effects that you would see for a typical steroid. And that clinical trial is expected to begin this year. So you're going to see quite a basis here of different approaches that I think will really inform the community, help you think about what you want to do and how it fits in with these mutation-agnostic approaches.
Great. I think our job is done now,
I am done.
Except for welcoming Jonathan Blaze to the stage.
Thank you all.
All right, good morning, everybody. It is a privilege to kick off this session, and I'm John Blaze, Field Medical Lead at PTC Therapeutics. And I want to thank the organizers at Cure Duchenne for giving us this opportunity to talk a little bit about PTC, our commitment to the Duchenne community, our continued support of Emflaza. I'm going to finish with some words about our white glove patient services program, PTC Cares. PTC is a public company. I'm going to share information today that is reflective of our observations and forthcoming expectations. However, outcomes may differ from what is expressed this morning. And for folks who might be unfamiliar, PTC is a global biopharmaceutical company that is focused on the development and commercialization of innovative and clinically differentiated therapies for individuals living with the highest unmet medical needs. The company was founded almost 30 years ago around the discovery of Adalurin, a read-through agent for the treatment of nonsense mutation DMD. In fact, PTC helped pioneer the study of Duchenne, conducting the first clinical trials in the space back in 2004, and bringing Emflaza, or deflazacort, to the US in 2017. A standard of care therapy, Emflaza was the first, and until recently, the only corticosteroid approved for the treatment of individuals living with Duchenne ages 2 and older. It comes in 2 formulations and is associated with 20 plus years of research, nearly a decade of commercial availability in the US, and a substantiated body of evidence supporting its efficacy, tolerability, and safety. This is all underscored, by the way, by nearly 20,000 person exposure years. Now, supporting this growing body of evidence, forming that body of evidence, are more than a dozen studies, inclusive of randomized, placebo-controlled clinical trials, large natural history data sets, and long-term, real-world observational studies that capture information of individuals living with Duchenne at various stages in their journey. In some, more than 2,000 people have participated in these studies, and many of whom have had more than 10 years of follow-up. Now, what you see here is just a subset of the studies that have informed the totality of evidence for Emflaza, and I'm going to share some of those with you today, beginning with the pivotal phase 3 randomized placebo-controlled trial that led to the approval of Emflaza in the United States. Here, 196 boys and young men with Duchenne, ages 5 to 15, would have their muscle strength measured, assessed. And what was demonstrated were that those on Emflaza would see a statistically significant preservation of muscle strength at week 12 when compared to placebo. This was the primary endpoint, and this is what led to the approval. But if you go further out to week 52, you see that those who were treated with Emflaza would have a statistically significant preservation of muscle strength when compared to prednisone. Now, these data are some of the first to really underscore the importance of early initiation corticosteroids for that protection of muscle, but also continued use of therapy even throughout loss of ambulation, for the preservation of muscle and the preservation of functional ability. They gave me a laser to call some things out, but it's really powerful, so I'm going to avoid using it to try not to set the screen on fire. Moving on to some more data around the preservation of that functional ability, we have the SYNERGY study. Of course, the SYNERGY study is the largest prospective observational natural history study in Duchenne. More than 440 participants, boys and young men, ages 2 to 28, followed for up to 10 years across 20 centers internationally. And what was seen was that those who were treated with Emflaza would benefit from a statistically significant delay to the time at which they lost key functional ability. And some of that included a 2-year delay in the time at which they lost the ability to rise from a supine position or rise from lying flat on the floor. So in other words, those who were on Emflaza would preserve the ability to rise from that supine position for an additional 2 years when compared to those on prednisone. There was a 2.7, nearly 3-year delay in the time to loss of ambulation. Obviously, loss of ambulation is a very important key milestone, and there are prognostic reasons for that, but more importantly, independence and quality of life. Those on Emflaza preserved the ability to walk for an additional 3 years compared to prednisone. And finally, a 2.7-year, nearly another 3-year delay in the time at which they lost hand-to-mouth function. Now, obviously, all of these contribute to the constellation of functional abilities that underpin independence, but more importantly than that, they translate to improved quality of life. And yet another study, this one a post hoc analysis of participants in the placebo arm of the ACT-DMD study, where these participants would be on standard of care. They would receive either prednisone or Emflaza. And what was seen over 48 weeks was that those who were on Emflaza would have a statistically significant and clinically meaningful preservation of walking distance, about 37.6 meters, about 125 feet, as measured by a 6-minute walk test. Now, these benefits were also seen in individuals living with Duchenne who, unfortunately, are not well represented in clinical studies or real-world studies. In the PRO-DMD study, this time a longitudinal cohort analysis, 86 young men who were non-ambulatory were shown to preserve this hand-to-mouth function in a numerically greater fashion when compared to no steroids. And this was true for either prednisone or deflazacort, or Emflaza. But when compared, or when looking at median time to age at loss of ability to turn aided in bed, it was only those who were on Emflaza who would see this statistically significant preservation, where those who were on Emflaza, when compared to prednisone, would have this preservation for up to 6 years. Again, the ability to turn unaided in bed, a key functional ability important for independence and important for quality of life. In the same study, regardless of corticosteroid, boys would see a statistically significant preservation of cardiac function, which is fantastic, but only those who were on Emflaza would see a significant preservation of lung function, a 2.3-year delay in which they went below a 60% forced vital capacity when compared to those who were not on corticosteroids, and about a 1-year delay to the same milestone for those who were on prednisone. Now, across these studies, deflazacort was better tolerated than daily prednisone. Patients who took prednisone had reports of increased weight gain and behavioral side effects. In other studies, including a real-world study, those who switched from prednisone to Emflaza would report fewer side effects, including less weight gain. And more recent studies have suggested that the improvement in these behavioral side effects may be attributed to the fact that Emflaza does not cross the blood-brain barrier as readily as other corticosteroids in the class. Now, it's important that I mention here that in the pivotal trial and across studies, boys treated with Emflaza have a tendency to form cataracts and have fractures. This is certainly captured in the prescribing information. It's obviously something that needs to be discussed with your doctors. But a recent study, a study from this year, a real-world observational analysis of 3,200 cases across the 9 years, spanning the 9+ years that Emflaza has been commercially available, suggests that reports of these adverse events are very low. In fact, cataracts was reported in less than 5% of all AEs are reported, and fractures in less than 1% of all AEs reported. So I think the take home here is that Emflaza has a very well-established safety profile with predictable adverse events that have known and well-studied management strategies. I am out of time, so I'm going to quickly summarize by just emphasizing the importance of this very robust body of evidence. Again, more than 20 years of research, about 10 years of commercial availability in the data therein, and nearly 20,000 person exposure years exist from Emflaza, again, underscoring its safety, tolerability, and efficacy. I'd be remiss if I didn't mention again our white glove patient services program, PTC Cares. This is a program that was designed to help support individuals living with Duchenne. We have several amazing case managers who've spent their life's work in Duchenne, some of them more than 25 years. We're still supporting low out-of-pocket costs and copay assistance. We're still offering bridge programs for individuals who are concerned about treatment gaps, and we offer investigation support for prior authorizations to get started on medication, reauthorizations to remain on medication, and for those who want to come back to Emflaza. The number is there, but feel free to reach us throughout the conference if you have any questions. Finally, I want to thank everybody for their time and attention. I want to apologize for definitely being over time here, and certainly call out all of the people whose lives have been touched by Duchenne who made these studies possible. I speak for all of the companies here that the strides that were made in this community would be impossible without your commitment and your sacrifice. So thank you very much.
Much appreciated. And let me bring up Dr. Schoenig from Catalyst to talk about their approach.
Good morning, everybody. I want to start by saying what an honor and a privilege to present in front of this group. And also, thank you for the opportunity to speak about the drug that we work on, Agamree, vamorolone. So my name is Alka Schoenig. I'm a physician by training, internal medicine, practiced for many years, and now really focused on patients with Duchenne muscular dystrophy. I represent Catalyst Pharmaceuticals Medical Affairs Department, and I want to start by saying thank you for being here. We know that living with Duchenne or caring for someone who has Duchenne is not a small thing. It shapes your world. It shapes everything that we do, the research and development we've done, the studies that we're doing now, the long-term data and follow-up. All that is oriented towards you and the quality of life that can be improved. We'd also like to recognize and thank Cure Duchenne and other advocacy organizations who have supported the development of vamorolone and Agamree over the decades. So thank you very much for that. So let's start with something of which all of you in the room already know, firsthand and deeply personally. Corticosteroids are a foundational treatment for Duchenne. My previous presenter spoke about it as well. They can slow progression by reducing inflammation and helping muscles function for longer. But we also know that for many families, the side effects also influence your adherence, your experience on the drug, and therefore it's very important for us to understand how side effects and the benefit profile can lead to meaningful clinical use of this medication. So corticosteroids can essentially slow the progression of Duchenne muscular dystrophy by reducing inflammation as well as helping muscles function for longer. And so we, in a study that was done in 2019, found that 6 out of 10, 60% of patients stopped taking corticosteroids because of side effects. Now that's really not a good risk profile for a drug that's a foundational therapy for Duchenne. So here's our drug, then I can speak a little bit about the mechanism of action. Agamree is thought to work by attaching to specific targets in the body, which then helps reduce the activity of proteins that play a key role in inflammation. So essentially, it works on both glucocorticoids and mineralocorticoids by suppressing inflammation and also having MR effect on antagonist effect. I think I'll go to the next slide because this is the one where we've really tried to simplify how we talk about the benefit and the risk profile of the drug. Essentially, we did a study as a part of the clinical development program, where we looked at the time to stand velocity. How fast could boys stand up from lying on their back, and how many rises could they do per second? And so if you look at the screen, there's a nice infographic there that shows that by 0.06 rises per second faster was the impact of vamorolone. And these results were maintained for 48 weeks. So the initial study was 24 weeks, and then there was an open label follow-up because this is a long chronic disease and you really don't want to stop with the short-term data. And so for 48 weeks, these patients were observed and the same benefit was there. So this is very important. The study was not, however, designed to do the comparisons after 24 weeks because it was a single arm extension. So that's the important part I do want to emphasize. And before I go back, I also want to say that this study was not set up for labeling or other indications, and essentially it was the longitudinal follow-up of the same group of people. So side effects are really important. We cannot talk about benefits without talking about side effects. And like other corticosteroids, vamorolone may cause serious side effects, including hormonal changes, behavioral changes, infection risk, blood pressure or salt changes, stomach and bowel problems, mood changes, bone thinning, eye problems, and vaccine-related risks. It's important that you talk to your healthcare provider when you see these before you start the drug. And I've also listed the most common side effects, and you can see them on our US prescribed PI, which is available also at our stand in the booth for Catalyst. Essentially, in terms of the side effects, they were similar to those seen at 24 weeks. So the long-term adverse event or safety profile was very similar to the 24-week safety profile. And another question that we often receive from families who are on prednisone, and which we really wanted to bring it up here, is what happens if we switch? Because most of the patients have started on prednisone for various reasons historically. And so what happens? So in this small study, when the boys were switched from prednisone to Agamree, certain side effects were noted lesser in the Agamree arm, in the vamorolone arm, including immune-related, behavior-related, and digestive side effects, as well as puffiness of the face and weight gain. These observations came from comparing the same patients across two studies, and so this was not a head-to-head comparison, and that is a limitation of the study, but the direction of those findings is essentially meaningful, especially clinically. And we also noted that after switching to Agamree, the boys were having normal growth velocity, showing an increase in the height percentile, which is very important because our patients are doing better, the quality of life is improving, the mortality and morbidity numbers are improving, and normal growth is really essential for our patients and how they perceive their important needs. In this next slide, I want to bring the attention towards the science is one part of the story, right? The physician will understand your patients, give you a prescription, but what happens after that? There's this whole overwhelming universe of getting this new prescription in your list of medicines. How is that pathway going to work? So we have something what we call Catalyst Pathways, which is set up essentially as I might call a white glove concierge service, to really put together the entire flow of things around you that can help you get your medicine in the right way consistently and also educate you. So parts of that include first, access and reimbursement support. It lists here free bridge medication and low out-of-pocket costs. Most importantly, it provides educational programs on what can you expect as your disease current diagnosis, how is it going to progress, what can you do to improve your healthcare outcomes, and also ongoing personalized support. And I'm very happy to state that Matt, whose quote is presented here, is present at the Catalyst booth. So he would love to hear from you. And his quote essentially is, "It's a privilege to help patients and families through disease education and navigating the complexities of the healthcare system." Well said, Matt, because that really captures what the experience is for the patients and their families. And essentially, Catalyst Pathways helps us support you and your families in that journey. Important safety information. I think I've covered some of this, but it is a prescription medicine that is used to treat Duchenne in patients 2 years of age and older. And if you should not be taking vamorolone if you are allergic to it or any of the inactive ingredients in the medicine. So again, a list of side effects here. Do not stop the medicine or change the amount taken. Increased risk of infection, increase in blood pressure and water retention, hole in the stomach, severe behavioral and mood changes, risk of osteoporosis, cataracts or glaucoma, and when you get vaccines, you need to get healthcare provider review. You need to inform them that you're on this medicine because there are considerations. Other important considerations include hepatic or liver issues if you're pregnant or planning to become pregnant or breastfeeding, and also certain medications. So make sure that you mention to your provider that you are on other medications, and then they can make sure if there are any drug-drug interactions or concomitant medications we need to watch about. I have mentioned the potential side effects as well. I do want to start, end and start both by saying thank you for allowing us to be your trusted partners. And we'd also like to share that we are doing long-term extension and follow-up for the use of AGAMREE in these patient populations. SUMMIT is the name of the study. It's our 5-year prospective registry study, which also has some active information gathering. And it will help us show in the real world how the drug actually works, not just through clinical practice, but really collecting data in a very standardized manner that we will then be presenting, of course. So if you want to learn more about this study, please come to the Catalyst booth. We have our healthcare and advocacy colleagues who can help you understand more about that study. Thank you so much for your time, your trust, and for allowing us to be a partner in your journey. Thank you.
Excellent, thank you so much. Thank you, everybody, and thank you for that. Next up is Jay Chauhan from ITF. We're going to hear the latest news on Duvizat. Thank you so much. Jay?
Hey, good morning, folks. So my name is Jay. I'm here with ITF Therapeutics. I've been in this space for 4 years now, and every time I get a chance to give a talk like this directly to patients, family members, caregivers, honestly, it's the most meaningful to me. I think a lot of you will agree that in the past few years, a lot has changed, and that was reiterated this morning by Deborah. But important progress has been made, and there's still a lot more to learn. So today, I'm excited to share with you what all we've learned with our long-term safety data, hopefully in a way that feels useful and relevant to you. And this is a roadmap of where we're headed in the next several minutes, but I want you to take away today with who is ITF, what is Duvizat, and who is it for, what we've learned from our clinical trials, and our ongoing commitment to this community. Just a little bit of background of ITF. We are the US arm of a broader company called Italfarmaco, and as the name suggests, they're based in Italy. We're based just right outside of Boston, and we're a relatively new organization. I like to say we're lean but focused. We were founded in January of 2024, and shortly after, we received FDA approval of Duvizat in March of 2024. Since then, we've continued to be collaborative with this community, honestly learning just as much as we share, and that partnership is something that we take very seriously. Here's some important safety information listed for you here. These are some side effects that patients may experience on Duvizat, and there's some important warning and precautions I should mention according to our label. Some patients may see low platelet counts or thrombocytopenia. Some patients may see an elevation in their triglycerides, and some patients may also experience diarrhea and vomiting. Throughout this presentation, I'm going to highlight some of the more common side effects that we did see in our trial. And today, this brings me to a short disclaimer. Folks, I am not a healthcare provider. You all are being well cared for by care teams across the nation. So before making any medical decisions for yourself or your family, please, I urge you to consult them. So what is Duvizat? I asked my toddler this, and he said, that's a Pokemon. And so if you think I'd like to clarify that for you. Duvizat is a non-steroidal treatment indicated for Duchenne muscular dystrophy. It's for people aged 6 years of age or older. and is designed to be used regardless of your genetic mutation or your ambulatory status. It is an oral suspension taken twice daily, and I mentioned it's not a steroid. It belongs to a group of drugs called histone deacetylase inhibitors, or HDAC inhibitors for short. And stay with me a moment for the next slide. I'm going to explain why targeting that pathway makes sense and a drug like Duvizat could be useful. So let's take a quick step back and understand one of the pathways how muscle repairs itself. In that process, we use proteins are used called HDACs, and they play a key role in how muscles repair and maintain themselves. In Duchenne, that system becomes overactive and it's unbalanced. This overactivity leads to constant muscle damage, and what you see is more inflammation and your repair system becoming really overwhelmed. The idea behind a drug like Duvizat is to calm down or reduce that overactivity of HDACs, therefore allowing your body a chance to breathe, honestly, and an opportunity to repair itself instead of being in a constant cycle of inflammation and fibrosis. Which brings us to the next important questions. What does this look like in people living with Duchenne? This was our pivotal phase 3 randomized double-blind placebo-controlled trial. It's a lot of words. In simple terms, 2/3 of patients received Duvizat, 1/3 received placebo. Keep in mind, in both groups, all patients received steroids, and this trial lasted 72 weeks or about 18 months. In total, 179 patients were enrolled, once again, age 6 years of age or older, and dosing was based on weight, and adjustments were made needed for tolerability. At the end of the trial, participants did have an option to continue into our long-term extension. So let's look a little deeper into the design of the study. I think there's some interesting things here because this is where it starts to become meaningful. One of the main measures we looked at was a 4-stair climb test, and that measures both strength and function. Some of the additional measurements we looked at that you may be familiar with is the NSAA. We assess muscle strength directly looking at movements like bending of the elbow or straightening of the knee. And finally, we wanted to look at visually, how does muscle change over time? And we'll get to that in a second. These are the results showing you for our main measure, the 4-stair climb test. If I direct your attention to the left, this is showing you the change over time and how long it took participants to complete this test. And I think on the right, it summarizes it quite well at the end of the study. Patients treated with Duvizat showed a significant less decline, and that's depicted by patients completing the test 1.78 seconds faster versus placebo group. And I mentioned we looked at some additional results. I'll start with here on the left. What you're seeing here is MRI scans of your thigh muscle, specifically one of the largest muscles in your quadriceps. And these are scans of 2 individual patients. The top is a patient taking steroids alone, and the bottom is a patient taking Duvizat and steroids. And the white area, or the light gray area, represents fat tissue. And if you look at those 2 patients, if you compare both of those from the beginning to the end of the trial, I think you can clearly see that the patient that was taking Duvizat and steroids showed less accumulation of fat over time. And folks, I know this is just data from 2 individual patients, but it really helped us illustrate what's going on at the muscle level. On the right, you'll find the additional measures that we looked at, and in summary, all the measures favored Givinostat over placebo. With any therapy, safety is a top consideration, and it is a challenge to capture every single adverse effect that happens in a clinical trial, but some of the more common ones that occurred with our participants were diarrhea, nausea or vomiting, abdominal pain, low platelet counts, and an increase in triglycerides. I do want to mention that these effects can be monitored over time through routine lab tests and clinical follow-up. And those guidelines are really in place. And if you have any questions regarding monitoring or management, I advise you to please talk to your clinical care team. All right, so that was EPIDUS. After EPIDUS, I mentioned participants could engage in a long-term extension study. And this study it's important to mention, this study is separated in a couple of different ways. One, there's no placebo group. And 2, the main goal is to understand how Duvizat performs over a long period of time on a safety perspective. This study included 194 participants, once again, age 6 years of age or older, And at this moment, all participants are on Duvyzat for at least 18 months, and some participants have been have been followed up for at least 8 years. When we looked at the safety results, they were consistent with the EPIDA study that I mentioned. Most patients did experience side effects. However, a majority were mild or moderate in severity. Serious side effects did occur, and 12% of participants. Importantly, none were fatal, life-threatening, or considered related to treatment. I mentioned the main goal of this long-term study was looking at safety. I think equally as important, researchers found that it was important to measure how key everyday functions change over time. And I like this slide. I think it really brings the data to life. What we found was, looking at the results, that it suggests that participants that are taking Duvyzat and steroids, on average, were able to maintain the ability to rise from the floor 2 years longer, ability to climb 4 stairs 3.3 years longer, and maintain the ability to walk 2.9 years longer. So that was EPIDUS, our long-term trial. Our commitment doesn't end there. I like to think in many ways this is just the beginning. What we value as a company is your feedback, your insight. How can we design better trials? And this is what we've done. We've taken your feedback and building on what we already know. This includes studies like the Providus study. This is looking at how Duvyzat is used in the real world. We know in the real world it's much more complex. How does Duvyzat work alongside other therapies in both ambulant and non-ambulant participants? Or our DMD voice study, hearing directly from caregivers and patients and families, what does this look like on a day-to-day basis? Study 52, this is gonna really help us understand safety and how the body reacts to the medication in our younger participants, age 2 to 6. And finally, we have the Ulysses study, which is our international participants, strictly the non-ambulant group. I've shared a lot with you in a little amount of time, but what stays with me most is the people behind the data. None of this would be possible without the participation, the involvement of investigators, this community, and your feedback is so important. You only see me on stage representing ITF so far, but there's several of us here. Please, if you see us, come say hello. It does not have to be about Duvyzat, just get to know us and know that we're here for you. And with that, I'll say thank you. I hope you enjoy the rest of the session. Take care.
Thank you, Jay. Much appreciated. On stage now is Joanne Donovan. Joanne's the Chief Medical Officer at Edgewise, and we're going to get an update on sevasemten, their experimental medicine in phase 2 and phase 3 trials. Joanne. Yeah.
Thank you, Mike. It's such a pleasure to be here at Cure Duchenne again. Really delighted to work with Cure Duchenne. We've been supported by them for years and early investors, so it is great to be here. So today what I'm going to speak about is our approach, as you heard from Mike, to preserving muscle. And we are a public company, so this is our forward-looking statement that I'm going to talk about things that may or may not happen in the future. And importantly, sevasemten is an investigational agent and it's not approved in any territory. So Edgewise has been on a mission to discover new medicines to improve the lives of people facing serious muscle disease. We've been around since 2017, and we're the only company that's actually focused on the proteins that do the muscle contraction. And our goal is to develop an investigational agent, sevasemten, that is taken as an oral pill daily to preserve and protect muscle, to be used either alone in combination. And one of the important features is that it can be used with any mutation. So what's the fundamental problem? You know that the lack of dystrophin or the partial lack of dystrophin in Becker is the problem. So what does dystrophin do? In the left here, the healthy muscle contracts, but it isn't all the muscle fibers contracting at once. And what dystrophin does is it links the muscle across the muscle, the different fibers. And so when one muscle fiber contracts, it's actually supported by the other muscle fibers. And on the right, when dystrophin is absent, that muscle fiber is all by itself. It's all alone. And if you think of the way a rope works, it's got all little fibers that are individually easy to snap, but when you twist them together and give them that lateral support, the rope is very strong. So that's how we're trying to address the fundamental problem of dystrophin not working well and protecting the muscle. So there are particular fibers called the fast muscle fibers that are somewhat more susceptible, and those are the ones that we are targeting. And sevasemten does this in the fast muscle fibers. We have clinical trials that are ongoing, and we have both in Becker and Duchenne, and I'll tell you a little bit about them. Now, this is a electron or a visible light microscope vision of a finger muscle of a mouse. It's small enough we can see it and see through it. So on the left is what happens, the MDX mouse, which is a model for Becker and Duchenne, and you can see those blebs happening. That's akin to those individual muscle fibers snapping and being damaged. And the novel idea that our founders identified was if you can decrease contraction just a little bit, so the muscle functions fine, does what it needs to do, but you take the edge off of it, then it completely protects against that visible contraction damage and the other biochemical evidence of damage as well. So what we've done is we started in Becker muscular dystrophy. We've done several trials, and all of those trials lead into an open label extension, which is now almost 4 years of patient exposure. And that trial is called MESA. I'll show you some data from this across the trials. We have a trial, Grand Canyon, that is a phase 3 essentially trial in Becker muscular dystrophy, and that will be reading out later this year. And we will have, at that point, the information that we hopefully need to go to FDA to go to approve that drug. So that's where we are in the Becker program. Now, I'm actually gonna skip one here, and it looks funny. But basically, let me walk you through this slide. This shows the North Star ambulatory assessment of the patients in the original trials, and then they went from the placebo-controlled trial over to MESA in the long-term extension. So this shows 2.5 years after they started on sevasemten. The vertical axis is the North Star score, and horizontal is time. The triangle there shows what the natural history studies have shown, that there is decline in the North Star scores in Becker. We have 50 patients at a year. We have 24 patients at 2 years, excuse me, more than that, 30. And what we see is that they're stable, that we've preserved function in these patients, which is really exciting. And so that's why we're very hopeful about the phase 3 study. We've also, we obviously look at safety. This represents many years of overall patient years, and there haven't been significant safety signals. The most common adverse events are overall in the trials are dizziness and somnolence, and those tend to happen in the first few days of treatment and then disappear. So it's been very well tolerated. 99% of the patients in the trial have continued on into the open label extension. So in Duchenne, we've started 2 trials. We've started LYNX in boys that are on corticosteroids largely, and potentially if they're on exon skipping. FOX, we included boys who had previously been on gene therapy. And we are looking at the landscape here. We're looking at how things change. We need to know, and there's going to be a session later today, how do the combinations work? Can we layer these things on top of one another? So the idea was to understand what the dose is, what the population of patients we should be treating in a future study. And this was to determine that, and what to do that, we looked at a range of doses. We looked at, and this is a schematic of the trial, based on what we had seen in Becker, we did 12 weeks to look to see if they were biomarker changes. And as I don't need to tell this group, clinical trials in Duchenne are hard. And we do them to figure out the next steps. Well, we didn't see the same biomarker changes that we saw in Becker, but we did later on. And because we had changed the doses, that made it difficult to sort out the actual signal. We also saw, and we heard from parents, that the higher doses were not as well tolerated. So we moved everybody back to the 10 milligram dose. And after a year, we saw, and we presented last year and spoke about with Mike at a Cure Duchenne webinar, that we were seeing stability, encouraging stability over that time. And we also have looked at boys who started later to understand if they were only on that dose, is that beneficial? So in summary, we are planning to share more data next month. And what we are seeing is that functional observations support our 10 milligram dose. We're trying to understand which patients are potentially more benefited by this based on the mechanism of action. And we are continuing to treat the boys in the open label extension. So to summarize, my thank you slide disappeared, I guess. There we go. But just to thank you, to thank all of the families, the boys, the men that are involved. We know that clinical trials are not easy, and we do try to do what we can, everything we can, to make it easier for them, minimizing visits. But ultimately, it still rests on the community to get these trials done and to make sure that we have the information to move forward. So, many thanks.
Thank you. Okay, next up is Mark Awadallah. Mark is from Capricor, and we're going to get an update on their late-stage completed phase 3 product that's going in front of the FDA this year. This is Deramiocel, and there's some really interesting and exciting data that Mark's going to share.
Hello. How's everybody doing today? Oh, this is great. Okay, Mark Awadallah, I'm the Chief Development Officer for Capricor. So Capricor has been in the development in Duchenne for a cell therapy for the past 20 years. And back this past December, those 20 years of work and research finally came to fruition with the readout of the HOPE-3 trial, which was the pivotal phase 3 trial that looked at the safety and efficacy in patients with Duchenne muscular dystrophy who are late ambulatory, non-ambulatory patients over the age of 10. So these are our forward-looking statements, a bunch of stuff that the lawyers tell me I have to put up. I'm not going to talk about it because I don't know what it says, but here you go. So, like I said, we read it out, and what does that mean? We applied, essentially submitted all our data and our clinical study report to the FDA, and the FDA said, we are going to make a decision on your application on or before August 22nd. So everybody say a prayer, light a candle, burn some incense, because this is a very important day for Capricor. What is Deramiocel? And so right now, let's start with having some audience participation, all right? What is Deramiocel? So everybody, if you're like me, hate the names of drugs. I can never pronounce them. So when we were trying to figure out what the name of this drug should be, we kind of landed on something called Deramiocel, right? So I want everybody to repeat after me, Deramiocel. Deramiocel. So it's not so hard. So I think we did a good job with that one. So what is deramiocel? Deramiocel is not a stem cell therapy. It's not a stem cell at all. What it is, it's a very rare cell population that's found in heart muscle. And so what Capricor does is that we actually get human hearts that were intended for a heart transplant, but never made it to fruition for some reason or another. And believe it or not, that happens about 40% of the time. And so we have a telephone, almost kind of like the bat phone, that we get a call in the middle of the night, hey, there's a heart that's available, do you guys want it? We go through some criteria that we review, and then we get it in-house. Once we get it in-house, we start processing it and finding those rare cells in that heart muscle, and then we end up growing out those cells, and we infuse it. And we infuse it to patients every 3 months. And we've been doing this for quite a while. We've had some patients that have been on the studies for over 5 years, doing pretty well. And what do these cells do? Well, these cells have a mechanism of action that is antifibrotic, and that's very important, and we'll talk about later why. It's immunomodulatory and anti-inflammatory. So that's what these cells are intended to do. They don't replace dystrophin, they don't make dystrophin, which some of the exon skippers kind of do with the truncated dystrophins. They're not stem cells, they don't engraft. They get infused, they send out signals to be antifibrotic to the other cells and anti-inflammatory, and they go away. That's why we have to infuse every 3 months. So this is the data. We had a HOPE-3 study, which was ongoing for a couple of years now, and we had 106 patients that were enrolled, and they were randomized, half on placebo and half on deramiocel. The primary endpoint was the performance of the upper limb, because like I mentioned, most of the folks that participated in the trial were non-ambulatory. We also looked at cardiac endpoints called left ventricular ejection fraction. The stuff is made from a heart, and we wanted to see if it helps the heart. Okay? Overall, this slide pretty much talks about the patient population. And what's important is that the balance between placebo and active was overall generally balanced. That means we had the same amount of patients that were on average the same age as placebo and active and so forth. Overall, the safety results were favorable. Most of the adverse events were mild to moderate in grade. We had one grade 4 life-threatening event that was on a placebo patient. And overall, the drug was well tolerated. And that's what about the adverse events. Most of them were headache, runny nose, itchy throat. They lasted about 24 to 48 hours and essentially resolved. And this was the expected profile that we've seen since the inception of all our clinical trials. So this is just an overview of our endpoints. And it's funny, there's this thing called type 1 error-controlled endpoints. It's a very fancy statistical term. And what does that mean? So when we do a clinical trial, we have to essentially name the endpoints before we read out. And we say, okay, these are our endpoints. When we say it's type 1 error-controlled, that means that they're in a hierarchy. That means in order to get credit for one, you have to hit the one before it. So the first one has to be statistically significant before you get to the second one. And the second one has to be statistically significant before you get credit for the third one, and so forth. If you miss number 2 and you go to number 3, and even that's significant, you're never going to get credit for it because it missed the hierarchy. So what we're trying to show here is that these are all the 5 endpoints that were in that order, and they were all statistically significant. And what statistical significant means is that it's the probability that a measure did not happen by chance. So for us, we had to hit a threshold of 95% probability for that endpoint not to have happened by chance. And so anything below 0.05 is a good thing. And if you see here in the red box, everything was statistically significant. Our primary endpoint was the performance of the upper limb. And that was a statistical significant endpoint showing a 54% slowing of disease. And so the difference between active and placebo was about 1.2 points on the performance of the upper limb. And what we did is we measured things by the percent change of the PUL 2.0 and not the raw change, right? So the raw change is 1.2, but what is the percent change? Well, we know that this is a very heterogeneous patient population. We have patients that were as old as 22 coming into the study and patients as young as 10. Well, all those patients are on a different trajectory in their disease course. Well, how do we make that the same? So how do we analyze it the same? So think of it this way. A patient comes in and their performance of the upper limb, they had a starting score of 40. And they lose 2 points over the course of the year. We had another patient that entered the trial, and their starting score was a 20, and they lost 2 points. Well, do those 2 points mean the same for that patient that started as 20 compared to the patient that started as 40? It's totally different. And that's why what we did is we said, okay, no, we're going to look at the change for each patient and then average it out instead of just taking a raw number. And that's what we did to analyze this. And we saw that statistically, patients that were on deramiocel were better off over the course of a year. Our key secondary endpoint was an endpoint that had to do with the heart, the left ventricular ejection fraction. And left ventricular ejection fraction is the heart's ability to pump blood through the body. And so overall, we saw a statistical significant positive outcome whereby patients declined 2.4 percentage points of LVEF over the course of the year compared to patients on active treatment, which equated to about a 91% slowing of disease. And that was all the patients that had analyzable data. So some patients had cardiomyopathy to start, and some patients didn't, right? So we said, well, what if we just looked at those patients that already had cardiomyopathy? And so this is what we saw. So we saw a better effect of deramiocel in patients with cardiomyopathy. So that means that they had some documented cardiomyopathy in their medical records. Their starting ejection fraction was below 55%, or they had evidence of scarring on their cardiac MRI. And we saw here that the patients had overall stabilization that were on deramiocel compared to patients on placebo. So we also looked at something called late gadolinium enhancement. And all that is that it's a contrast media that enables us to see the scar around the heart and if there's fibrofatty infiltration. And so we saw that patients that were treated with deramiocel had overall stability in the progression of that scarring compared to patients on placebo. Placebo patients increased in the number of segments of the heart by 3 compared to deramiocel, who did not see essentially any increase in scarring. And these pictures are pictures that would show on the left-hand side in the blue box was a patient that was on placebo, on active treatment on deramiocel. They started out with those yellow arrows that showed where the scarring infiltration happened into the heart muscle. And at month 12, it was the same scarring that was there when they first started the study. And the one in the red box was a placebo patient. It showed that they had a little bit of scarring on their heart, but over the course of 12 months, that scarring really increased. So we also did something called a global statistical test. All right, so we wanted to find a way to quantify something of how a patient feels, functions, and survives, right? Those are terms that the FDA likes to use a lot. Well, how does the patient feel, function, and survive? So what we did is, we tried to put that into some sort of calculation. So we took every patient's data from their PUL 2.0, from their left ventricular ejection fraction, and something called the patient global impression of severity. So the patient global impression of severity was a measure that represents how a patient feels. And the performance of the upper limb was how a patient functions. And the left ventricular ejection fraction is how a patient survives, because we all know that cardiomyopathy is the leading cause of death in Duchenne. And so we took all that data and combined it, and what came out is that we saw that the patients on deramiocel were statistically significantly better off than patients on placebo for this kind of calculation of how a patient feels, functions, and survives. And then we have something called the mid-level PUL. We all know that the performance of the upper limb is split up into 3 domains: your shoulder domain, your mid-level domain, and your distal domain. The mid-level domain is probably one of the important ones because it really, helps patients with their independence, their ability to feed themselves, their ability to brush their teeth, their ability to reach out and grab their phone. And so we also looked at that domain alone, and we also compared it with the Duchenne Video Assessment. So this is an assessment that was done, and if anybody participated in the HOPE-3 trial, they would take videos of the patients, and one of the assessments was their ability to eat 10 bites, and they measured essentially their ability, and then at every time point. And after the 12-month time point, we see that both the mid-level domain and the Duchenne Video Assessment of Eat 10 Bites, which is essentially very similar in their function. We wanted to make sure that what we're seeing is we can replicate it. And you see the trajectory of the patients on placebo is very similar in both of the endpoints compared to deramiocel. So both ways you look at it, we're showing statistical significance in these 2 measures. So, in conclusion, right, the HOPE-3 study met every one of its endpoints, which was a huge win for us at Capricor, and hopefully one day it'll be a huge win for the community once it's approved. We see that deramiocel slows down the progression of the loss of skeletal muscle function by measurements of the PUL 2.0 and the Eat 10 Bites. From a cardiac perspective, we saw that LVEF the progression of disease was significantly slowed down in all patients that we had MRIs for, especially in patients with cardiomyopathy. We also showed that we've stabilized the progression of late gadolinium enhancement, which is the scarring. And remember how I told you that the mechanism of action is anti-fibrotic? Well, the scarring in the heart is essentially fibrosis. And so this endpoint was a very big win for Capricor because it proved our mechanism of action. We're saying that we are anti-fibrotic. Well, here's the data to prove it from an imaging perspective. And yeah, that's pretty much all I have. HOPE-3 confirms our hypothesis that deramiocel is beneficial from both musculoskeletal and cardiac functional benefit in patients with DMD. And with that, I really, on behalf of Capricor, want to thank all the families and the caregivers and the patients who really dedicate their time, their efforts into participating in all these clinical trials, because without the patients, there would be no drug development at all. And also, Cure Duchenne, Deborah, and all the great people at Cure Duchenne for all their hard work. Thank you so much for everything you guys do. That's all I got.
Well done. We're going to stick with the same theme. We've got TJ Singe from Secretome, and again, this is going to be on their stem cell, cardiac-derived stem cell approach, and this one here is going to be slightly different. Thank you, TJ.
Hi, good morning and good afternoon. We're getting there to lunchtime. My name is Teji Singh from Secretome. Thank you so much for inviting us here. I just want to introduce Secretome to you since this is our first presentation at Cure Duchenne. Our method obviously here is to preserve, protect, and regenerate muscle. When we say the heart of, it's because that's the source of our material. Basically, what are we as a company? We're a clinical stage biotech company. Our lead program is STEM-01, so much easier to pronounce than the previous, and it's for DMD-associated cardiomyopathy. We're actually already in the clinic for DCM, dilated cardiomyopathy, and heart failure with preserved ejection fraction. So this is our foray into the Duchenne space. Now, everyone's already spoken about the disease. Joanne did a great job explaining what happens in the absence of functional dystrophin. So what basically we see is that you have multiple areas of muscle, skeletal, cardiac, diaphragmatic muscle that's going to be damaged. And the purpose of the secretome that we have here, which is the same thing with other stem cell type therapies or cell therapies, is to modulate that damage, reduce inflammation, prevent damage from fibrosis, enhance repair, potentially also help to strengthen the muscle fibers themselves. STEM-01, our compound, is basically neonatal cardiac progenitor cells. This is cells that are collected from the right appendage of the atrium during cardiac surgery in neonates. So these are kids under 30 days of age. They're undergoing a surgical procedure, usually for a congenital heart disease, and when they're having that procedure done, a small bit of their right atrium tissue is taken out as part of the procedure to put in a cannula. We take that tissue, and then we take it to our lab, and we can actually propagate it into these lines of tissues that we call STEM-01. The advantage of this one is it's a neonatal source of tissue, so it doesn't actually have the same issues that you have sometimes with adult cells. Adult cells aren't as viable, don't produce as many copies. Adult cells may also have some immune markers that in the neonate under 30 days you don't find. And you can actually see that when a kid cuts themselves or hurts themselves, their scar disappears really easy. When I cut myself, my hand is all messed up for a long period of time and it's got this horrible keloid. So we have a study that we're planning to start in DMD. We've already submitted the protocol to the agency. We're just waiting for some feedback from them. It hopefully starts this summer. It's going to be a study in 20 male participants, age 8 and up. They can be ambulatory or non-ambulatory. They do have to have evidence of cardiac involvement. And then what we're going to do is very similar to what our colleagues at Capricor have done, is we're going to be infusing these cells every 6 weeks. So one of the differences here is that a little bit more frequent, but the cells will actually survive that period of time, and then the body will eliminate them. The study is an open label study. It's going to be done with dosing in the clinic, and if everything goes well in the clinic, we're going to be able to set up at-home dosing. If everything there goes well in this study, because the primary function of it is to actually understand safety. If we understand that the safety is good, we don't have issues with any sort of reactions, obviously that makes it possible to bring it to the home, to the infusions there, but the safety is our first key aspect. And then the things that we're doing actually for efficacy assessment, very similar to our colleagues, we'll be looking at left ventricular ejection fraction, pull, quality of life. Those measurements will be done at 12 months, and then the study itself will be going on for at least 18 months. If that study goes well and we get positive data, we are in parallel are designing our phase 3 study. We're having discussions with the agency now about the design of it, and that's why we're wishing Capricor great luck with that PDUFA date, because the results of that will be really important to the field, the community, and will also help us really understand what we have to study. So when we get that result back with the agency, we'll finalize our phase 3 design. That phase 3 study would be our registration study. That'll be a global study, not just in the US, same population, but again, similar endpoints to what's already there. So I was going a little fast because I don't want you guys to miss lunch and keep everyone on track. But yeah, just thank you so much for inviting us here. We're really looking forward to working with the community, and obviously the patients, the investigators, and Cure Duchenne are the reason that we're all going to be making Duchenne a lot better place to be. Thank you.
Thank you, TJ. I appreciate that. Up next is a totally different company. It's going to be Satellos, talking about their opportunity of muscle regeneration. We've got their Chief Medical Officer with us today, Wildon Farwell. Wildon, the stage is yours.
Hello, everybody. Thanks for sticking with us. I'm Wildon Farwell. I'm the Chief Medical Officer at Satellos, and I'm very happy to be able to present to you. And thank you to Cure Duchenne for hosting such a great conference, and for all of you for being with us. I am the Chief Medical Officer at Satellos, and we're going to be talking today about SAT-3247. This is an investigational therapy not yet approved. As the other speakers, we have lawyers at Satellos who require us to put legal disclaimers about forward-looking statements. So at Satellos, we're really founded upon this key question. Why does muscle regeneration decline over time in people living with Duchenne muscular dystrophy? We believe that if we can answer this question, that we may be able to understand new therapies and new approaches to people living with Duchenne and other serious muscle diseases. So many in the room are very familiar with the natural course of the Duchenne muscular dystrophy. You know that when babies are born and as they grow, they are able to develop function. They are able to learn to sit, to stand, to walk. But then there's this period of time where that begins to regress. They're not able to maintain the function that they've had. And the question is why? What is going on? What has happened so that the body is unable to keep up with all the muscle damage that is occurring? Well, what you've heard many times at this conference and in previous presentations is the classic understanding of the role of dystrophin, and that's as a structural protein within the membrane. What's recently been discovered, Michael Rudnicki, one of the founders of Satellos, discovered that dystrophin has a role not only in the muscle fiber, but also within the satellite cell, the stem cell within muscle. And when dystrophin is missing, those stem cells are not able to divide in the same manner. They're not able to create muscle progenitor cells. And when that's missing, the muscle's not able to keep up with all of the damage that occurs. So if we walk through this a little bit more, what you see on the left, or on, yes, your left-hand side, is you see the healthy muscle in a non-diseased person. You see that the muscle is able to create what's called polarized state. It's able to create this asymmetric division where muscle progenitor cells are produced as well as stem cells. But in people living with Duchenne, where dystrophin is missing, these stem cells aren't able to polarize. They're not able to divide in the same way. They create more muscle stem cells. This has actually been seen in biopsies of people living with Duchenne for many years. People weren't able to explain this. What is going on? All these stem cells, but yet they're not able to make more muscle progenitor cells. What Michael Rudnicki discovered is that there are backup mechanisms that can be drugged to enhance the stem cell polarity. And so he developed an oral small molecule, SAT-3247, to investigate whether inhibiting a protein called AAK1 could enhance stem cell polarity and therefore muscle regeneration. So Satellos has conducted a phase 1 clinical study. We're now expanding that study. The first study was CL101. There were 5 adults with Duchenne that were enrolled. They completed 28 days of dosing. I'll present some data from that study here in just a second. We've now started Trailhead. Trailhead will include participants with Duchenne from that first study. 4 of the 5 have returned and are continuing to be monitored and treated with 3247 in that study. We're also expanding that study to include 25 additional adults between the ages of 16 and 25. We'll have additional sites in Australia as well as sites in the US. And so We have initiated conversations with clinical trial sites in the US, and hopefully we'll be able to enroll here soon. But if we just step back to what were the results that were seen in that phase 1 study, they were very encouraging results. What was seen was that in the 5 adults, 3 of the 5 had a marked improvement in hand grip strength. In the total, about a doubling in hand grip strength over 28 days. Again, short period of time, small number of participants, but very encouraging data. And what was also noted was that the participants who did the best were those participants who had more muscle mass when they started. Again, consistent with the biology of how we believe the drug will work, you have to have muscle that has stem cells, they want to divide, then 3247 can allow more of that division to occur to create more muscle progenitor cells. So in Trailhead, the follow-up to that study, we recently reported some new data. What we saw was, again, a continued safety profile that is unremarkable, meaning we did not see significant concerns. 2 participants reported adverse events, none of which were considered serious or related. We saw that the hand grip strength that was initially observed in the 101 study was maintained, and in some participants actually continued to improve. We also began to evaluate strength across new muscle groups, so across the elbow and the shoulder, and we saw stability, and in those participants who had more muscle mass, there was actually some improvement in those muscle groups. We also noted that pulmonary function appeared to be stable for most participants. There was 1 participant who had some variability in their respiratory function, but overall, we believe that these continue to be encouraging results and make us look forward to what's to come. So the new study that we are enrolling now is BASECAMP. BASECAMP is our phase 2 trial. It's a placebo-controlled study in boys between the ages of 7 and 10. 3-month placebo control, followed by a 9-month open label extension. We'll be evaluating muscle biopsy, muscle MRI. We'll be looking at different strength and functional assessments, biomarkers. We currently have 11 sites open in the US, Canada, Australia. We're looking to open additional sites in Europe. We are on target to fully enroll the study within the 3rd quarter, which should then give us the opportunity to evaluate data starting in the 4th quarter of this year. So in conclusion, at Satellos, we believe impaired muscle regeneration plays a key role in Duchenne and other muscular dystrophies. We know that dystrophin is a structural protein, but dystrophin is also important in stem cells and satellite cells. And when it's missing, these cells lack the opportunity to regenerate. So 3247 is designed to inhibit AAK1 to then allow better cell polarization, to then allow asymmetric division, to then create more muscle progenitor cells to allow the natural process of muscle repair and regeneration. So far, early days, small data, but it's encouraging. Safety profile looks reasonable thus far, and we're excited to continue to advance the program with Trailhead and BASECAMP, and we look forward to presenting new data later this year. And again, thank you everyone, Cure Duchenne, all of the families here, everyone in the community. We could not be doing this without you, and we look forward to participating with you over the next few years. So thank you again.
Appreciate that. Yeah. The final one today is Ken Acker. He's from Grunenthal. I got that right? Yeah. So, I mentioned at the beginning, many of you may not have heard of Grunenthal before. They're developing Seagrams, these selective glucocorticoid receptor molecules that are going to hopefully have similar properties and better potency than steroids, but without the metabolic side effects. Grunenthal, being a European company, has got slightly different rules and regulations. So Ken is coming out and he's forced to read off the computer word for word as per European regulations. So it's a slightly different format. And so the floor is yours, Ken.
Thank you. Thank you. Thank you, everyone. Appreciate the opportunity to be here and speak to you about Grunenthal. We are new to Duchenne. Me personally, I've been in the Duchenne space now about 3 years, launching a previous product for another organization. So again, thank you to Cure Duchenne for inviting us to provide an update on our clinical program. We're honored and humbled by this opportunity. Grunenthal is a research-driven pharmaceutical company headquartered in Aachen, Germany. Although Grunenthal is newer to the Duchenne community, our purpose has always been simple, and that's to improve the lives of patients. I'll apologize up front as I don't have any data like my colleagues have all presented data. We're early on at Grunenthal. We just completed a phase 1. We're moving into a phase 2. So our goal is to obviously publish our preclinical data over the next year and then provide more data on our phase 2 when that's available. So several available FDA medications target the glucocorticoid receptor and have demonstrated improvements in muscle function, slow muscle degeneration, and delay progression in the DMD. Without curative treatments available in DMD, long-term glucocorticoid steroid therapy remains standard of care, but is constrained by dose and duration-dependent side effects. Families and clinicians must constantly balance benefit and risk, pursuing the essential goal of preserving muscle function while managing unwanted side effects. Selective glucocorticoid receptor agonists and modulators, also referred to as Seagrams, they're investigational compounds designed to engage the glucocorticoid receptor in a more targeted way. Research is exploring whether Seagrams may influence inflammatory pathways with less side effects. However, the clinical implications of this mechanism have not yet been established. Steroids bind to the glucocorticoid receptor inside cells, influencing gene expression. Research describes 2 main patterns of activity. That's transrepression, which is linked to anti-inflammatory effects, and transactivation, which is associated with metabolic and growth-related side effects. Seagrams are designed to interact with the glucocorticoid receptor with the goal of influencing receptor signaling in a way that may emphasize anti-inflammatory pathways while reducing activation of other downstream pathways. In theory, this is an appealing approach. In practice, the biology is quite complex. Inflammation control and metabolic regulation are interconnected. The receptor does not operate in isolated pathways. Over time, research has shown that fully separating benefit from risk is challenging because these signaling pathways often overlap. So what is Grunenthal doing? Grunenthal is developing an investigational Seagram designed with novel chemistry. In preclinical models, including the MDX model, which is widely used, a genetic model used in Duchenne to characterize by mutation in dystrophin gene that leads to muscle degeneration. We observed that in this investigational compound was associated reductions in diaphragm inflammation and improved muscle force. Whether these findings translate clinically remains to be evaluated. Remember, this is all preclinical data, so we're not sure how that will translate to humans, but hopefully we're super excited to forward our program in phase 2 and then move hopefully on to phase 3. So our clinical development plan for our investigational product compound, as initially mentioned, we completed phase 1. We'll commence our phase 2 study in 4 to less than 7-year-old boys with Duchenne who have not previously been on steroids. The centers have been identified in Europe and US. These are some of the study locations. Oops, let me go back. We've identified some sites in the US as well as globally across the country. This is a nice picture of our campus in Aachen, Germany. I don't have a pointer, I don't believe, but we actually do have labs there. Grunenthal is a very old, established company, and this is where all the research takes place in Aachen, Germany. So we want to thank Cure Duchenne, the families, the clinicians, and the researchers in driving progress, especially to the patients and families participating in clinical trials. Without your support, we would not be able to advance science. Progress in Duchenne depends on collaboration, community. We're proud to be a part of this community and mission. Thank you so much.
Thank you, Ken. Appreciate that. I want to just thank every one of the presenters in the session this morning. Incredibly informative. You can see quite a diverse array of different types of approaches here. Some of them at late stage, getting ready for PDUFA dates, some of them at earlier stage. But this has guided the community into what's out there as you think about therapies and how you want to make decisions around them. So I just want to thank every one of them again. Right. I know that we're right at lunch, and I just want to remind you that I will be back on stage at 2. We've got a great session on both gene therapy and gene editing, and there's going to be some very interesting science that I encourage everybody to be able to be here and understand essentially what that means for therapy today as well as going forward. So thank you all.